In this video, we'll be looking at DNA technologies. And by DNA technologies, I mean, I guess, techniques and methods that are used in a lab with molecular biology when working with DNA. So our first step is we need some DNA to start off with. We need some DNA to work with. So we'd get some cells first. So it depends what kind of DNA we're looking for. If we're just getting DNA from bacteria, we'd grow up some bacterial cells and harvest them. If we want kind of human cells, we might just do a cheek swab to get some cells from our cheek. So we'd get our cells first and put them in a test tube. Then we need to add some chemicals to that that's going to break up the like the cellular membrane, the membrane of the organelles, for example. So it's going to disrupt those membranes and we'll be left with just a kind of a goopy mix of just all cellular constituents just kind of mixed together. So by that, I mean, there'd be some DNA, probably RNA, proteins, lipids, carbohydrates, all just in this kind of goopy mess. So the next step after kind of breaking up the cells is we need to then start separating out these cellular constituents depending on what we need. So in this case, we're looking for DNA. Now, this is all done basically just by I guess, a few repeated cycles, based on kind of one simple principle. And what I mean by that is you would then add a solution, and the solution you'd be adding would be based on what you're trying to actually separate out. So you'd add a solution where some parts are going to be soluble in that solution and other parts are not going to be soluble. The next step would be you would put your tube in a centrifuge, which kind of spins the tubes around really fast. And that means that the insoluble parts, after they're the heaviest, they're going to then kind of be packed down tightly at the bottom of your tube, and the soluble parts are going to be kind of then left in the liquid. So just showing you a couple of pictures of that. This is just a picture of a centrifuge. You'd put all your little kind of tubes there, close the lid, turn it on. It then kind of spins around really, really fast, and all of the soluble parts will be left at the bottom. So if we look at these tubes, let's kind of like forget about this label. This one on the right may be what something may look like when you've added a lysis buffer, for example. Like by a lysis buffer, I mean the solution to break up all of the cells. You kind of mix it up. So that's what it looks like before. Then you might put it in a centrifuge, spin it down. All of the insoluble parts are going to be kind of packed down tightly at the bottom of the tube. The soluble parts will then be left in the liquid. Notice that it's not packed down exactly at the bottom because the tubes are put in on a bit of an angle. So now you've separated these two parts, you then just are going to decide what you want to keep. So if you wanted to keep, for example, what is dissolved in the liquid, you might use a pipette to kind of suck up all of the liquid and transfer that to a fresh, clean tube. And then you could just throw away this packed down pellet at the bottom. Or if, vice versa, if you wanted to keep this, you might just pour off all the liquid to get rid of what's soluble in the liquid, to be left with only the pellet. So once you've done this kind of one round of separating things out, you would then add a different solution and mix it up. And in that different solution, you would then have more constituents that would be soluble in the liquid, and other parts that are insoluble. You would centrifuge that again, decide which part you're going to keep and which part you're going to throw out, add another solution. So you'd keep repeating that until you'd be left with only one thing. And in this case, the DNA. So this is what it would look like at the end. So the DNA, is often a white pellet you'll find at the bottom of your tube after kind of centrifuging it down. So this is a very small tube. That's someone's finger. So DNA is a white pellet. This is actually quite a bit of DNA. Often your DNA pellet could just be like a bit of a tiny little spot that you might only just see. And you would then kind of discard that liquid. You would add in like, I guess, a special buffer where DNA is just nice and stable and happy in, and you can then kind of store that DNA in solution in the freezer and pull out whenever you want. So that's what you'll get at the end. So our next little technique we'll talk about is the polymerase chain reaction, or PCR. So this is used to amplify DNA. And by amplify, what we mean is, I guess, just get more copies. Because often when you start this process, you'd be starting with just a small amount of DNA, which can be hard to work with. So you would use PCR to make lots of extra copies of your DNA, so you can then have a lot more to work with. So before we go into, I guess, PCR and how it works, we just need to understand primers. So now you may remember when talking about DNA replication, that one of the important enzymes is DNA primase. And it's used because it kind of provides a little bit of a starting off primer on the second strand of DNA. So then DNA polymerase can come in and start synthesizing the second strand. Now, keep in mind that DNA polymerase cannot work if there's no little kind of primer starting it off. So if we just had this, this blue single strand and we didn't have a primer at all, DNA polymerase can't do anything. So it needs some little bit to kind of start it off. And that's basically what a primer is and what DNA primase provides in DNA replication. Now, in the case of PCR, pardon me, in the case of PCR, you can kind of design and choose specific primers to be amplifying up just one little section of DNA. So I'll show you what I mean by that. So let's say we've isolated our genomic DNA. So let's say that's the whole genome, but I've got my little kind of my favorite gene here, and I'll just put a second strand in. So I've got my favorite gene here, and that's the DNA that I'm interested in. I'm not really interested in any of these side bits. I just kind of want to work with this. So I can design specific primers that are going to be kind of complementary to there, and that kind of means that DNA polymerase is going to work along and synthesize on that strand. So I'm just going to choose a sequence that's just on this side of my favorite gene. Then I can just choose a little bit of a sequence that's on the other side of my favorite gene, another primer there. That means that DNA polymerase is going to work along that way. So by having a primer on either side of my gene, then that means I can, if I'm going to amplify my little bit of DNA, that means I'm only going to be amplifying this little bit. I'm not really, I'm not interested in any of these other bits. So I'll end up with kind of like lots and lots of copies of just this little bit of DNA that I'm interested in. And as I said, I can kind of define the boundaries of what I amplify by just where the primers are that I'm deciding to use. And I can decide all of that because we know the sequence of all of this. We can just look on a computer at the sequence of our gene, choose a sequence that's on either side of our gene to kind of use for our primers. So I'll just go back to sharing. So that's essentially that picture that I kind of drew simply is basically what this is showing. So if we kind of say we want to, the part of the DNA we want to amplify is this little bit here, we're just going to kind of choose primers that are kind of complementary to a little bit of sequence on either side of the DNA that we want. So that's where we go down to here. So we've got one primer that's going to be complementary to here, so that DNA polymerase is going to work along this way. We'll choose another primer that's complementary to the other strand that's on the other side of the DNA we want to amplify. It's going to work the other way. So the end result will be is we're amplifying this bit of DNA only and not what's on either side of it. So we'll end up with lots of copies of our favorite gene to work with. So how does PCR work? Let's just kind of quickly go through the origins of it. You don't need to know it in this much detail, but I think this is a really good example of someone kind of putting a few kind of basic ideas together, or just a few basic tools and techniques, and actually coming up with something kind of really interesting just with some clever uses of basic tools. So it all started by isolating the DNA polymerase enzyme from a bacteria that's kind of very stable at high temperatures. So it's a bacteria that might live in thermal vents or thermal pools where everything's kind of very hot. And the optimum temperature for this DNA polymerase was 72 degrees. So the inventor of PCR kind of put together a few things. So this thing started it all off. We've got a DNA polymerase that will work happily at 72 degrees. Now we need our DNA primers. So we've spoken about that. So if we want to amplify up a specific little section of DNA, we can just design primers to be just on either side of our bit of DNA we want to amplify. Now the next little thing we need to think about is the highest possible annealing temperature. And I know that doesn't mean much to you now, but I'll draw some pictures to demonstrate. Now, if you remember earlier in the Year 11 course, we spoke about DNA hybridization. And so I'll do a couple of examples of, so I've separated some two strands of DNA, and I'm trying to get kind of like two different strands to kind of join together. So let's say if I've got some mismatches in the base pairs, so let's say those two don't match up, those two don't match up, those two don't match up. And let's say if I compare that with another sequence of DNA where everything matches up, so I've got these two options. Now, if I then, if I was to, for example, then just mix them together in the one tube and then I start heating it up slowly, just going up by 1 degree at a time, I am going to find that this one is going to melt at the lowest temperature because it's not held together tightly, because there are several base pairs where it's not actually joined. So there might be one temperature where this is going to then split apart because it's not held together tightly. But then I might have to go up a few more degrees, higher temperature, to then split this one apart, because we'll say that this one is going to be joined at every single base pair. So we can kind of extend that concept to, if let's say I've got my whole genome here, and I've designed a little primer, and so my little primer sequence is between 20 or 30 base pairs, and there's only one spot in the whole genome that matches exactly with that. So now if I try to have a temperature where my primer is going to join to my single stranded DNA, like if my temperature is too low, I might get my primer going here, and that might be where I want it to go. But if my temperature is too low, I might get my primer also binding here and also binding here with a few little mismatches. But if I raise my temperature a little bit more, I'll get to a temperature where that's not going to bind anymore because of the mismatches, and it's only going to bind in the one and only place in the genome where I designed it to go, which means that every single base pair kind of matches up. So if we can kind of choose the right temperature for our primer to actually join up to our genomic DNA, we can make sure that our primer is going to bind to that only one point in the whole genome. So let's go back to here. So yeah, so if we kind of just have the right annealing temperature, so the primer binds at only one place, and then we know also that we can kind of separate our two DNA strands at a high temperature of 95 degrees. So what does PCR do? So the beauty of it is we can add everything just to the one tube. So we can have our DNA sample, which is our genomic DNA we've isolated. We've got our primers that we designed. We need some free nucleotides for DNA polymerase to be adding to the second strand. We have our DNA polymerase that works at 72 degrees. Then we have all of this in a bit of a buffer mix, so our reaction can go ahead, and we can put all of that in the one tube. And this one tube can just go into our one little PCR machine, and we can go through the PCR reaction. So how that actually works, and this is just looking at one cycle. So if we start off with our genomic DNA that we've isolated, so it's double stranded to start off with, we're going to heat it up to very hot so that the two strands separate. Then we're going to cool it down to the highest possible temperature, where our primers will only bind to that one spot in the DNA where we want them to go. And that's often going to be in the range of like 50 to 56 degrees. So our primers are going to join up in the one spot we want. Then we'll go to 72 degrees, which is where our DNA polymerase works well, and we're going to then synthesize the second strand. So notice we've started off with one double-stranded bit of DNA there, and by kind of cycling through those different temperatures, and each of these stages we might only need to do for a minute or two. Then we end up with two copies of the DNA. So this is just one cycle. If we were to then do this again and again, the amount of DNA we would get would double each time. And that's kind of what this is showing. So this looks a bit different, but we're just kind of showing the same idea, where we've got one thing of double-stranded DNA. We go through one cycle, we're going to end up with two copies. We go through another cycle, we go from 2 to 4. Another cycle, we go from 4 to 8. Another cycle, we go from 8 to 16. So it's doubling every time. So usually a PCR, you might go through, let's say about 30 odd cycles. That's really all you need to then get huge amounts of DNA, because the growth is exponential. Now this kind of explains a couple of things, where you may have heard of PCR being used for testing for coronavirus. So they can use PCR to try to use primers to specifically amplify only DNA that is from the virus and not from the person. So if there is viral DNA in the person, they then may be able to, they would do PCR, and starting with just the tiniest little amounts. like, because theoretically we only need kind of like one DNA molecule in our sample to be able to detect it with PCR. We can amplify it up so it goes exponentially, doubles each time, and then we can detect it at the end. So that can be used in the tests we have when they kind of swab our throat and nose for coronavirus, but it's also been used in testing kind of wastewater and sewage in different areas, in Sydney and around New South Wales, where let's say you've got a country town just with one sewage treatment plant that might deal with a town of, say, I don't know, say 40,000 people. They have done routine testing of the sewage wastewater and been able to detect the presence of coronavirus, in a town of 40,000 people when no one in the town has actually had tested and they didn't actually know someone was there in coronavirus. So you can kind of think how sensitive that test is when you're looking at just the sewage from a whole town of 40,000 people, and they're able to detect if one person has coronavirus. Again, just because PCR can be very, very sensitive by starting with just theoretically one molecule, but it'll amplify it up to a big amount so it's then detectable. So what would we use PCR for? Well, just like a couple of things. So we can use very specific primers to kind of amplify just one little single section of DNA like we've shown. We could also kind of use kind of shorter non-specific primers, so primers that'll basically kind of bind anywhere and everywhere in the genome to kind of basically amplify a whole genome. There are also kind of fancier applications of PCR where you could use PCR to introduce a single nucleotide mutation into a gene if you want. You can add restriction enzyme sites onto the ends of your primers if you want to amplify a section of DNA and have a restriction enzyme at either side of that DNA, so you can then clone that into something else. But let's just worry about this and say PCR is just used for making extra copies of our DNA, so we can work with more. Okay, so the next little one is restriction enzymes. And basically, just as this picture indicates, restriction enzymes are just a little tool that we'll use for cutting DNA. So they're enzymes that will cut DNA at specific sequences. Now, why do cells have these? Well, basically, they come from bacteria where the job of these enzymes is to cut out virus DNA that has inserted itself into the genome. So, for example, what a virus will do if it's infecting bacteria is the virus will just inject the virus DNA into the cell, and that cell is kind of put into the genome of the bacterial cell. But the bacterial cell has this cool little defense mechanism where these enzymes are able to say, hey, this DNA doesn't belong here, and they'll cut it out. So that's where it comes from. And basically different species of bacteria each have their own kind of restriction enzymes that will kind of cut at their own kind of specific sequence of DNA. So this just kind of shows you one little example of how it works. So this is like the enzyme EcoR1 that comes from E. coli. So it recognizes a sequence of 6 nucleotides. So that's the sequence it recognizes, but where it cuts is kind of shown by the red line like that. So it cuts, and that's kind of what we're ends with. So we're kind of left with what's often referred to as these sticky ends, because we've got a bit of an overhang of where it could actually kind of stick on to another bit of single-stranded DNA. So that's just we don't need to know this specific detail or how it works. I'm just showing you how it works. And now that the next one, again, you don't need to know this detail or remember it, but I'm just kind of showing you that scientists can basically build up a library of different restriction enzymes. So these are all just enzymes from different bacteria. Each one has its own little sequence that it will kind of cut at. So the idea is once kind of scientists have built up kind of like a big library of different enzymes, then there's a good chance that they can try and find a sequence to cut it. So they might be saying, I want to kind of cut the DNA somewhere at this point, and then they can analyze that DNA, and they might be able to say, oh, it turns out there's a sequence for this enzyme to cut it, so I can use this enzyme to cut it there. Or they might find a sequence, oh yeah, this one here, there's that sequence is in the area that I want to cut, so I'm going to use that enzyme. Okay, so uses of restriction enzymes. So as I said, so scientists have built up a library of different enzymes from different bacteria. Each one cuts at a specific sequence. So by having their big library of enzymes to choose from, you can kind of choose the one that's going to cut at the location where you want to cut it. To do a restriction enzyme digest, you'll have your DNA that you want to cut. You'll have that like in a buffer solution. You'll add the enzyme, and then to just incubate that at the enzyme's optimum temperature. It could be for a few hours. It could be overnight. And then at the end, if you want to kind of check if it worked, you can then run your DNA on a gel, like using gel electrophoresis. And you should then hopefully have a DNA fragment that is the correct size where you wanted to cut it. So you'll be able to know if I cut it here and here, I should have a DNA fragment of X number of base pairs. You can check that at the end by putting that on a gel. So what is this gel electrophoresis that I've been talking about? I mean, you should hopefully have seen that in some of our worksheets. So it's used to analyze the length of DNA fragments. So that's just simply all it does. But it turns out there are a few applications for gel electrophoresis that you can use it for that are all just based on looking at the length of DNA. So we can run our samples on the gel. We can compare their lengths to a DNA ladder, which has samples with known lengths to determine the lengths of your samples. So this here is just an example of a DNA ladder. There's kind of more than one type you can use. So this is just one. So this kind of shows, firstly, looking on the right-hand side, like each band is a specific length. So I got this as, like 10,000 base pairs, 8,000, each one going down to 100. Now notice when you actually look at the ladder on the gel, each band is not the same brightness, and that's kind of done deliberately, kind of to help you find your way around and work out which band does which. But it also can be a handy little tool that when you run your sample of DNA, if your kind of band for your DNA is about the same brightness as one of these bands, you can just choose which band it's about the same brightness as. You can actually get a little bit of an estimate as well as to how much DNA is there, because you notice that there's more DNA in this band, which gives it a bright appearance than these other bands. So that's just an extra little thing you can use to estimate how much DNA you have. You can get a more exact thing using other ways, but yeah, so it tells you how long, but also can give you an idea of how much DNA you've got as well. So how does gel electrophoresis work? So the DNA molecule has a negative charge, and that's from the phosphate groups in the backbone. So that means if you have run electrical current kind of between negative and positive terminals, and you put the DNA in the middle, the DNA is going to move away from the negative terminal. It will move away from the like charge, and the negative charge DNA will move towards the positive terminal. So that's how we get the DNA to move. And now the next part is, how do we separate it based on size? So the way we do that is by making the DNA molecule have to kind of snake or worm its way through an agarose gel. That kind of slows down the longer strands. So if we imagine the agarose gel is like a big matrix, and the DNA molecule it's almost like it's got to go through an obstacle course. So the smaller strands are able to kind of move the fastest. You could say they're the lightest. They don't get kind of caught up or snagged as much. So the smallest strands can move very fast. The very longest DNA strands, which kind of are heavier, they kind of get snagged more. They move more slowly. So the end result of your DNA run on an agarose gel is the DNA bands that have traveled the furthest. They're the shortest. The longest strands of DNA have only traveled a small distance. There's just a few pictures kind of showing you what the kind of apparatus looks like. Firstly, let's maybe start here at the top right. So that's just an actual photo of what it looks like, but it's not really kind of clear what's going on there. So let's look at a couple of diagrams. So this one here in the bottom right, that's basically like a side view of just this plastic gel tank. It's not actually including the kind of like power supply. So we've got a side view of the gel tank. So the actual gel itself, like the DNA agarose gel, is just this green thing here. And it's kind of sitting completely submerged in a buffer. And we're going to have a negative electrode in the buffer on one side, a positive electrode on the other. So that just means that the direction of the kind of the current and the way that the DNA molecules will move is going to be moving this way. And we've got our little well, which is basically like little kind of holes in the agarose gel where we'll put our DNA sample. So once it starts moving, it starts off in this little hole, but then it kind of moves into the gel, and then it's moving inside the gel as it goes across. So that's kind of what it looks like from the side. this kind of shows you then a bit more of a 3D image. So we'll have several wells along the top that we'll load our DNA samples into. Then when we turn the gel on, that's going to move along. So if we have DNA fragments in our sample that were all different sizes, then we're going to have the shortest ones will be, have traveled the furthest. The longest bits of DNA will have only traveled a little bit here. So a little bit more about about why it moves that way. I've got, firstly at the top right, I've got this little kind of picture just to kind of try to show that the lightest, smallest DNA molecules, let's imagine they can run the fastest. Whereas the molecules that are like the DNA molecules that are very long, we could say that they're heavy, they can't run so fast. The ones in the middle, they'll be in the middle. So the idea is with the gel is like this matrix, so we can imagine it maybe like a sponge that just has a whole bunch of holes that the DNA molecule has to kind of snake its way through, or you can imagine it's like a big giant jungle gym it's got to try to worm its way through, but basically it's this kind of porous matrix that the molecules have to find their way through. And the idea is that the smallest molecules, molecules of the same size will all travel at the same speed. So the smallest ones will all travel fast, but also the smallest ones that are all the same size travel together. So you'd end up with small ones of the same size will all travel about the same distance. Molecules that are a little bit bigger, they get slowed down a little bit. And then the very biggest DNA molecules, they get slowed down a lot and they can't travel very far. So that what this looks like on the gel is these kind of bands that we get. So this kind of band that we get here is that this means that there's made up of DNA molecules that were all the same size. They've all traveled the same distance. That's why we have a band. Same with these two. So we've got this band because the molecules were all the same size in a band. If we had molecules of every different possible size, we might just have like what's called a smear. Like, so rather than just one little distinct band, it's like we just had a paintbrush and kind of like painted all the way across if we had DNA molecules of every possible size. But here there was only this size put in that well, put in well number 3. Only this size was put in well number 2. DNA molecules of only this size were put in well number 1. So what do these gels look like in real life? So these are just kind of several examples. This kind of shows you what the DNA ladder looks like. They've got a ladder on either side of the gel. There's another DNA ladder there, DNA ladder there, DNA ladder there. Now, so we may see a single band just based on what we're loading, or if it was, for example, say a restriction enzyme digest of genomic DNA, we may see several bands from the one sample. So that just kind of shows you what it will look like. So let's just quickly go through all the steps in making and running a gel. So first of all, making the gel, it's much the same as if you were going to, I don't know, make some jelly. So for example, you're going to add your agarose to a buffer solution. You're going to heat it up so it dissolves. You'll pour it into a mold and then allow it to cool. So as I said, pretty much like you would just do if you're making jelly. So the next thing is we want to add a gel loading dye to your DNA sample. So this kind of does two things. It's a kind of a thick, heavy liquid that makes sure your sample sinks down into the well, because remember, the well is completely submerged in the buffer. We don't want it to all just dissolve and float away. So that makes sure that our DNA sample sinks down into the well. But the loading dye also gives an indication as to kind of how far the DNA has run. Because keep in mind, we can only see the DNA bands when we look at the gel under UV light, because the DNA is labeled kind of with a fluorescent label. If the gel isn't under UV light, we can't see those DNA bands. So the gel gives us, I guess, just one little visual indication that's a bit of an estimate as to how far the DNA might have run before we put it under UV light to really check. So we'll add the gel loading dye to our DNA. We're then just going to load the mixture of gel loading dye and DNA into the wells. We'll make sure to include a DNA ladder so we can kind of compare the lengths if we want. Actually, how about I'll jump back because I didn't kind of specify that, but I mean, hopefully it makes sense. So let's use this one because I've got numbers. So with our DNA ladder here, if we want to determine the length of any of these bands, like let's look at this band, for example. I want to know how big that is. I'll refer back to see how does that kind of compare with the DNA ladder. And it's kind of showing it's between 400 and 500 base pairs. So I don't know, it's not exactly halfway, it's a bit closer to 500. So I might guess, I don't know, let's say that could be, I don't know, 470 base pairs roughly. That's basically what you'll do. You'll pick a band and then you'll kind of refer it back to see where it fits in with these known numbers, and you can estimate the size. Yeah, so we'll run the gel, and as we know, the shortest DNA fragments will travel the longest distance. Then at the end, we want to visualize the results. So as I said, we can only actually see the bands when we look at it under UV light, because we need to add a fluorescent label. Now this fluorescent label could have been added to the actual gel at the beginning when we added the agarose and kind of heated it up to dissolve it. So it could be present in the gel, or after the gel has run. Sometimes people will remove the gel from the tank and soak it in the solution for a while that contains that fluorescent probe. So once we've done that, we'll then just put it on under UV light and take a photo and get our results. So applications of gel electrophoresis. So there's quite a few. So it can be used in DNA profiling, where you have done a restriction enzyme digest on genomic samples from different people, and you're trying to compare the bands to see how many bands actually match up and how many bands don't. It can be done to check if either restriction enzyme digest or a PCR has worked, because if you've done a restriction enzyme digest, you've hopefully cut out a DNA fragment of that's X base pairs long, and so you can check that on your gel. Same with PCR. With PCR, you might be trying to amplify a section of DNA that is X base pairs long, and so to check that actually worked, you can put a little sample of your results on a gel to check if it worked. You can also use it to purify DNA. You might even want a specific band from a sample where you might have a DNA sample where there are that gives many bands, but you might be interested in just one band. So you can physically run it on a gel and use a little scalpel or knife to actually cut out one band from your gel, put that in a test tube and kind of dissolve it so you've basically purified just one little band from your gel. And then, we can use the DNA ladder, as we said as well, to give a bit of an estimate of DNA concentration. So there's kind of lots of things we can use a gel for, and this doesn't even cover all of it. So summary, this is just the very, very basics kind of to take away into what these techniques are used for. So remember our DNA isolation, we need to get a pure DNA sample and have it separated out from all of the other cellular parts. PCR, we'll use that to make more copies, or typo there, that should be copies of your DNA, so you have more DNA to work with. Restriction enzymes, they kind of will cut DNA at specific sequences. And DNA electrophoresis is used to analyze DNA based on the length of DNA fragments. So what's an example of putting all of this together? So if we were looking at DNA profiling, for example, so we would isolate our DNA from our test subjects. We would amplify the DNA using PCR just so we have more to work with. Then next we might cut the DNA using the same restriction enzyme. It's important we use the same one because we're comparing different people. So we'll use one restriction enzyme on everyone. We'll then analyze the length of the DNA fragments using gel electrophoresis. So when we're then looking at the different bands on our gel, any differences we see in the length of DNA fragments we see on the gel would be due to the different numbers of repeated sequences kind of between the places that we've cut. So if there's more repeated sequences, that would give us a longer DNA fragment. If there's shorter, a smaller number of repeated sequences, that would give us a smaller band for that bit of DNA where we cut it. So the fewer differences we see between different samples, then we'd say the more closely related the two test subjects are. If we see lots of differences, then we would say they're not very related. If they're identical, then that could be used in crime scene analysis, where we could say we have identified the suspect.